Hi, good morning, good afternoon, good evening, everybody. A very warm welcome to all of you joining us for this ICH webinar dedicated to the updates from the EHA ASCO 2024 annual meetings in myelodysplastic syndromes. I'm Mohamed Moti from the Sorbonne University and Saint-Antoine Hospital in Paris, in France. And it is my great pleasure to moderate this live webinar that will be delivered to you by Professor Amer Zeidan from Yale University in the US. Dr. Zeidan is really a top expert in this field, and we are very privileged and fortunate having him as the lead of the MDS and acute leukemia track as part of the ICH activities. And he has kindly accepted to deliver and to give you a sort of a best of what happened in the recent congresses in the field of MDS. And this is a rapidly moving field because we've seen even recently drug approvals, which is quite amazing, I think. So please do not hesitate to participate. It's an interactive webinar. Please share your questions. We'll have a Q&A session at the end. But for the time being, I'd like to leave the floor to Professor Zeidan. Amer, the floor is yours.
Yeah, thank you so much, Dr. Mohti. And this has been an amazing, I think, year and a half almost for MDS with a lot of updates. So I wanted to kind of really acknowledge all the amazing efforts by Dr. Mohti and the International Academy of Clinical Hematology, because reach of the academy has been amazing as I go and give talks across really internationally. I'm really pleased to see how many people know of the activities of the IACH, and I think the very impressive efforts that are being done to educate hematology worldwide. So in my summary post EHA and ASCO, I just wanted to note two things as I start. One is there are always many amazing abstracts and many presentations, but you could cover so much in a limited period of time. So I have chosen what are the most clinically relevant. So those are selected abstracts that I thought are important for clinicians to know about, because they are drugs that are either already in practice with important practical information, or they are quite late in development. So I did not focus on a basic on translational updates from those meetings, or on early phase development that is probably years away from coming to the clinic. And of course, there are always many other abstracts that we don't have time to cover, but I'm happy to take any questions at the end in any specific area that I have not covered. So those are my disclosures. I'm just going to start by a quick background about MDS, and I divided my presentation into three main areas: some updates about the risk stratification and the response assessment, which has seen important development in the last year and a half in terms of the introduction of the molecular IPSS and the new response criteria for high-risk MDS. Then we'll go into updates into management of lower-risk MDS, and finally we'll conclude with high-risk MDS, which has seen a presentation of unfortunately negative, but important two large phase 3 trials. So I think the issue of the risk stratification continues to evolve in MDS. As you can see in this slide, historically we have used clinical parameters such as blast count, blood counts, as well as the karyotype to using different tools all the way from the IPSS to the revised IPSS. But most importantly, we have started to include molecular data formally now in the molecular IPSS. But at the end of the day, despite the number of risk groups, we end up from a pragmatic and clinical point to group patients into either higher or lower risk MDS. Because the treatment goal for lower risk MDS is still focused on improvement of quality of life, minimizing the risk of cytopenias, limiting transfusions for those who are anemic. While in high-risk MDS, the goal is still on trying to modify the disease course and prolong survival. Of course, the evolving classification systems which in the year 2020 have culminated in the basically publication of two competing schema, WHO 2022, as well as the international consensus classification, have started to complicate, I think, the field a little bit. I'm not going to spend too much time on this because it has been extensively covered in other settings. But while there's a significant overlap between these two classifications for myeloid malignancies, including important differences, and I think one of the biggest issues that have been kind of becoming more prominent is in patients who have blast count of 10 to 19%, what we call MDS/AML in the ICC. And is that being managed more like an AML or like a high risk MDS, and what response criteria to use, etc. However, having two different competing classification systems was always going to cause confusion for patients about what they have, complications in terms of drug development, in clinical trial protocol development, etc. So there has been a strong push internationally, including by the consortium that I've been very involved in, the International Consortium of MDS. And you can see here a map of the current experts. This is a global expert think tank, continues to grow. It's available on Twitter for those of you who are interested to know more about the activities. But this consortium, which started in 2020, has launched a number of activities. And I think the most important initiative so far was the effort to try to suggest a harmonized project for the classifications. This has been presented in EHA and ASH last year. And again, I'm not going to go through the results because this is a little bit from last year. But the bottom line is that we combined the data set from Europe and the US of more than 7,000 patients, and we used hierarchical clustering as well as a modified consensus Delphi process to using the expert opinion for areas that did not have a lot of specific evidence backing them to suggest a harmonized schema. This work is currently submitted and under review in a peer-reviewed journal. Hopefully, it will be published soon. But I think the goal really is, regardless whether this system, the ICC or the WHO, whichever system is being used, is that we end up with one common language that all physicians will use, which I think is very important. And to the same, I think, point, the response criteria in MDS has also seen significant evolution over time. In particular, when it comes to higher risk MDS, we have long recognized that the failure of drugs to move from lower risk MDS, from early phase trials to late phase trials, probably is in part at least driven by using clinical response criteria that don't fully capture the clinical meaningfulness of the responses. And there was an important effort to try to get rid, for example, of MARO-CR, which is blast reduction without blood count improvement, as part of those response criteria. And this effort basically also was undertaken with a large global network. These criteria, this effort started in 2021 after 1.5 years of basically reviewing all the literature about response criteria, including a formal systematic review and also a modified Delphi process with experts across the world. As you can see here, this was published in Blood early 2023 in terms of focusing on high-risk MDS. And this is, again, a busy slide, but it's kind of a top-line summary of that paper, which is available in Blood. You can read it in detail, as you can see here down in the references. But some of the main changes were reducing the hemoglobin threshold in the CR from 11 to 10, because hemoglobin of 10 is sufficient for transfusion independence. And most of us believe that you don't need to get to 11 to have a clinically meaningful count recovery. Of course, this is combined with blast reduction to less than 5%. As I mentioned, we also got rid of MARO-CR without count recovery. But importantly, we introduced a number of categories, what we call less than CR or near CR. So those are responses where the blast count is less than 5%. And you have a count recovery, but not all the way to meet the threshold of CR, which is hemoglobin of 10, ANC of 100, and platelet count of 100 and ANC of 1,000. So we introduced complete recovery with partial count, complete response with partial count recovery, CRH, which reaches neutrophil count of 500 and platelets of 50, or CRL, complete response with limited count recovery, which includes CRL uni and CRL bi. In these responses, you are achieving the count recovery in one lineage or two lineages to the same threshold like CR. And I think we also, of course, introduce a lot of specified definitions of time to event and issues related to the practical use of those criteria. The reason why I kind of go over this is that there has been a number of abstracts that I think are important to highlight, validating these IWG 2023 response criteria focused on high-risk MDS. I think very nice presentation came from the Austrian group using the Austrian registry, which has many patients. As you can see here, more than 1,000 patients in this particular analysis presented by Dr. Plyer and her colleagues. You can see to the left that the value of the composite CR, as well as the value of the overall response rate by the new definitions, is very well validated, as you can see, with very nice separation in the adjusted overall survival curves. But I think very nicely presented to the right figure is the CR and the CR bi and the CR uni, which, as I mentioned, those are basically, I think, important responses because they are associated not only with blast reduction, but meaningful count recovery. And I think you can see very nice correlation with OS in that survival curve. But I think what was even more intriguing in that analysis from the Austrian group, which used very sophisticated statistical methods, as you can see here, is that the IWG 2023 appeared to perform actually better than the old criteria, the 2006, but even better than the ELN 2022 AML criteria. And this is, as I mentioned in the beginning, an open question of which criteria is better, especially for patients, not only with patients with 10 to 19% blast, but also in patients who have oligoplastic AML. And I think because in the ELN 2022, they still use this category of morphologic leukemia-free state, where you have blast reduction without blood count improvement. I do think that this is probably contributing to why you are seeing better performance, as you can see here with the IWG 2023. And I think this is important to continue to study in different databases. And there was also another abstract that also validated these response criteria using the Ascertain trial database. This is the phase 3 trial that led to the approval of oral decitabine. So I think there is a number of abstracts now that and previously published papers that consolidate the use of the IWG 2023 as the main response criteria, I think, in my opinion, to be used in clinical trials going forward. And another, I think, concept on the response criteria validation, idea of using blood counts. Many of you who treat patients with MDS, especially those who are not transplant candidates, in their, like, let's say, late 70s, early 80s, you give them HMA and they achieve blood count normalization, and you want to do a bone marrow, and the patient is asking you, like, why do you want to do the bone marrow? Are you going to do anything different? And probably the answer is no. So there are many patients who don't get subsequent bone marrow biopsies. And of course, in the current schema of responses, you need a bone marrow biopsy to assess the blast count. So a very interesting concept that also was kind of led by Dr. Plyer from Austria looked at what we call peripheral blood complete response. So this is like looking at normalization of the blood counts without looking at the bone marrow. And she had shown that this actually carries the same prognostic value as complete response with the traditional complete response. And what we have done actually within the context of this large Validate, which is a large database of more than 2,000 patients from initially from the US, but as you can see in the map, we have extended it to Europe and across the world. Of all of those patients had high-risk MDS, which was HMA treated. And what you can see here is that we have shown using the new response criteria that this peripheral blood CR significantly overlaps with the traditionally defined CR Here we use the hemoglobin of 10. But I think, again, this speaks to the importance of blood count normalization. And probably we can assess responses without necessarily doing the bone marrow, especially as we combine molecular data, which we know now there is very nice correlation between blood and bone marrow molecular data. All of this means that in the future, we might not necessarily have to do repeated bone marrows on our patients to assess response, which I'm sure, as you know, patients will be very happy not to have to undergo repeated bone marrows. So now we are done with those like initial updates and classifications, response assessment. I'm going to move to the therapeutics, and I will start with lower risk MDS. So this is like the traditional approach for the management of lower risk MDS in patient, in generally, at least in the US, where all of these drugs are available. I think in some countries, not all of them are available, but for lower risk MDS, we continue to use the most common problem is anemia. More than 80% of the time, most than half of those patients will be transfusion dependent. For those patients who have an EPO level of, again, less than 2 to 500, historically, the main approach has been the use of ESAs, as you can see in this figure. Of course, for the 5 to 10% who have deletion 5q, you could start with ESA, but I think most of us here in the US will start with lenalidomide for those patients. And as many of you know, luspatercept was approved in the second line setting after ESA failure in patients who have ring sideroblast. But last year, I've seen very important development in the form of the COMMANDS trial, which I will show you, which basically has put luspatercept really as a frontline treatment for patients with lower risk MDS who are anemic and transfusion dependent, who do not have deletion 5q. But most importantly, and just 3 or 4 weeks ago in the US, the imetelstat, the first-in-class telomerase inhibitor, was approved in the second-line setting after ESA failure in patients who have lower risk MDS who are transfusion dependent. And I'm going to show you important updates from both the COMMANDS as well as the Amer clinical trial that led to the approval of both of those drugs. So this is the Medalist. This is a pivotal trial that led to approval of luspatercept. It was a randomized phase 3 trial that looked at luspatercept versus placebo. And this was after ESA failure. Luspatercept, as many of you know, is a ligand trap. What it does, it removes ligands of the transforming growth factor pathway, which have been shown to activate the SMAD signaling, which leads to an effective erythropoiesis. So removing those ligands will improve the erythropoiesis and improve the anemia. And you can see to the left that the top line results from the Medalist have shown an improvement in the rate of transfusion independence of 38% compared to 13% with placebo. And that led to the approval of the drug. We have also shown in subsequent follow-up from the Medalist that the benefits of luspatercept are not just in transfusion independence, but most patients actually derive benefit even if they do not achieve full transfusion independence by improvement in their blood counts. You can see here that the hemoglobin will go up, and even in patients who have heavy transfusion burden, most of them will experience significant reduction in their transfusion burden. So it might not be a clean win, like if you have a patient who has 7 units of blood every 2 months and they go to 2 units of blood every 2 months, that is not transfusion dependence. But I think from a patient perspective and from health utilization and space in the clinic and not needing blood transfusions, I think is important. And then came the COMMANDS trial, which aimed to look at the use of luspatercept in the frontline setting, and regardless of whether the patients had ring sideroblast or not. So this was a large international phase 3 trial that was conducted in the COVID era, and it was actually quite impressive that we were able to finish it despite all the COVID challenges. Those patients had lower risk MDS, they were transfusion dependent, and they were stratified by their red blood cell transfusion burden, by the EPO level, and about having ring sideroblast. As I mentioned, you could go on this trial regardless of whether you had ring sideroblast or not. Patients were randomized to receive luspatercept or erythropoietin, and both drugs were escalated. Very important to remember that both luspatercept and erythropoietin alpha need to escalate to the best dose to try to maximize the chance of response in most patients. And the primary endpoint in this trial, which was not just transfusion independence, it was a mixed response of transfusion independence with a mean hemoglobin rise of at least 1.5 grams per deciliter over 12 weeks. And what you can see, this was a clear win in favor of luspatercept, which had a transfusion independence of around 60% compared to 35%. This was the full analysis. The interim analysis, which led to the approval of the drug in the US, was already published in Lancet. This updated analysis using the full trial analysis after all the patients had the chance to be observed for the primary endpoint was also previously presented and now is in Lancet Hematology and Press, should be published soon. I think you can see a clear win across all the categories. There has been an ongoing debate about the RS negative patients where the rate of transfusion independence does not seem difference between luspatercept and ESA. However, the duration seems to be better in patients who are RS, in patients who have received luspatercept. And the most recent updates that were presented both in ASCO and EHA this year, I'm going to show you some of those highlights, but we're certainly basically consolidate the clinical value of using luspatercept in patients with lower risk MDS with anemia. An analysis from ASCO that basically looked at the entire duration of the study. As I mentioned, we had the interim analysis, we had the full analysis, and this analysis looks at patients through the end of the treatment, not only through the first 24 weeks of treatment, which was the full analysis. And what you can see here is that the rate of transfusion independence keeps going up, so that now it's 70%, basically with the primary endpoint compared to 43%. But importantly, you can see that to the right side that many patients, as I mentioned, even if they don't achieve transfusion independence, will achieve significant reduction in their red blood cell transfusion needs, and they will achieve a delayed basically need for the first unit. Patients who went on luspatercept did not need a unit of blood almost until 5 months into the trial, compared to around 40 days for those who were randomized to EPO. And the last point I would make is that the duration of transfusion independence was actually quite impressive on the trial. So it was 2.5 years with luspatercept compared to 1.5 years with EPO, like a year longer with luspatercept. But importantly, when you account for the multiple periods of transfusion independence, it's actually the median duration is close to 3 years. So certainly, I think very important benefits. But what also was interesting is we observed some improvement in the platelet and the neutrophil count in some patients. This is something that we already observed in the second-line setting in the Middle East trial, but we also observed the same thing in the COMMANDS trial. So in my mind, this does not mean that you use luspatercept to treat someone with neutropenia or thrombocytopenia with MDS, but I think it consolidates the value of the drug in patients who have borderline platelet and neutrophil count recovery. Because for sure, you're not worsening those counts, you might improve them, but the main benefit, of course, continues to be in fixing the anemia. And I think we are trying to take these benefits forward by treating patients in earlier settings. The ELEMENT trial, which is currently ongoing, I think is a very, very important trial, because this trial is trying to look at patients who are not yet transfusion dependent. This is, I think, is important variation between the US and Europe. In the US, we tend not to treat patients until they are transfusion dependent and with deep anemia, while I think patients tend to be treated a little bit earlier in Europe with ESAs when they have symptomatic anemia. And I think this is the right approach, is not to wait until the patients have very deep anemia, because by that time, in my opinion, there is already significant cardiac modulation and negative impact that are probably long-lasting. So this trial is looking at the idea of using luspatercept in patients who have a hemoglobin of less than 9.5 versus EPO, with the idea of trying to prolong the time to which they become transfusion dependent, and of course, with a number of secondary endpoints. So this is a very important trial. I think if you have it nearby, try to enroll patients on it, because it could move the field forward beyond the question of luspatercept in particular, but as a concept in terms of treating patients who are not transfusion dependent. And the MAXILLUS trial is another trial that is ongoing. The trial in progress abstract was also presented in EHA 2024. This is looking at using the higher dose of the luspatercept, which is 1.75 milligram per kilogram, because we know from real life analyses that most patients will not respond just to the starting dose, and they have to be escalated to the 1.75. So here, this trial is testing the question, since this drug is actually quite safe, whether you can start just with the highest dose with the 1.75 milligram. So I think this is an important trial. It's testing patients both who are ESA naive and post-ESA failure, but it's a single arm study. It's not a randomized trial. Now, each time you have a successful drug, of course, you are going to have second-in-class agents. And one of the most advanced ones that we currently have is this drug, KAR-050. This has some differences from luspatercept. It's also a ligand trap, but it also acts on activin A. And this drug basically has been presented more than once now from this phase 2 trial. Dr. Diaz-Campillo provided the update in EHA 2024. And here, again, you can see a lot of numbers, but I think the bottom line is that this drug seems to have activity, whether you have ring sideroblast positive or negative, and it seems to have some durable responses as well as improvement potentially in platelet count. However, I think it remains to be seen how does this drug compare to luspatercept and whether it fully distinguishes itself from luspatercept. There are plans to have randomized phase 3 trials with this drug, which hopefully will answer this question. Now, moving from the transforming growth factor beta modulating agents to imetelstat, which has been the other big news in lower risk MDS. This is a first-in-class telomerase inhibitor that basically takes advantage of the increased expression of telomerase in malignant cells, including those of MDS, because of the high proliferative rate of these cells trying to maintain their telomere length. And the use of this drug will work by inducing synthetic lethality. And this has been shown in patients with lower risk MDS to lead to higher rates of transfusion independence in a phase 2 trial, which ultimately led to the randomized phase 3 trial, the Amer trial. This was presented ASCO 2023 as well as in EHA, and finally published in the Lancet early 2024. And as I mentioned at the beginning, led to the approval of imetelstat in the US as a second-line agent after ESA. So this trial randomized a total of around 180 patients in a lot of countries. Again, this is a trial that was quite impressive because it was conducted during COVID. Patients were randomized to receive imetelstat versus placebo with an endpoint of 8-week transfusion independence. The patient actually had to have a significant transfusion burden. So you had to have more than 4 units of blood over 8 weeks to enter. The deletion 5q was not allowed because of the use of lenalidomide in that setting. And this is a paper I mentioned that has been already published in Lancet for those of you who are interested to kind of see the full initial results. But what was presented in EHA and ASCO this year were updates from the from the Amer study. I think the most important updates related to the durability of the response of this drug. So the median duration of transfusion independence when you did intention to treat by the arms among the patients who actually responded, among the responders, the 40% who achieved transfusion independence with imetelstat, it was 52 weeks. However, the longer you stayed on the drug and you were responding, the longer was the median duration. So for those patients who subsequently remained on drug more than 1 year, there were 18% who remained on drug for more than 1 year, and their median transfusion dependence was more than 2 years, 132 weeks. You can see that those who responded at 8 weeks achieved transfusion independence were very likely to achieve 6 months and 1 year of transfusion independence, which means that if you are transfusion independent at 8 weeks, you had more than 65% chance of being transfusion independent at 1 year. So I think, again, this speaks to the durability of response to this drug. But what also was, I think, impressive is the increase in the hemoglobin, the degree of increase of hemoglobin in this trial, where the rise of the hemoglobin in the imetelstat arm was an average of around 4 to 5 grams. And that increase, as you can see here in blue, that increase was actually better with time for those patients who were on the drug more than a year with transfusion independence. You can see the continued hemoglobin rise, where they landed with a hemoglobin of around a median of 13, which is, again, quite impressive. And there was no detrimental impact on overall survival. Of course, in these trials, it's always very tough to show an overall survival advantage with the given the limited number of patients that we have that go into these trials. But as a safety signal, I think it's important to show that there is no detriment in OS that comes with the transfusion independence, which we did not see. And also an important safety signal is we do not see an increase in disease progression or progression to AML with the drug compared to placebo. So I think the main messages here is that while this drug can cause cytopenias, I think it's very effective. However, the rate of neutropenia and thrombocytopenia is significant. Around 2/3 of patients will have grade 3 or higher neutropenia or thrombocytopenia. So I think of it more like using HMAs. And there was an important clinical question is, well, what are you like making the patient transfusion independent for blood only to make them deeply neutropenic and to make them need platelet transfusions because they are so thrombocytopenic? So we actually conducted another analysis using the Amer data set, which was published in or presented in ASCO 2024. And we showed important things, I think, that corrected some of those misconceptions. The first point is that the median number of platelet transfusions on the imetelstat arm was actually 1 unit. So most of those patients were not like needing basically a unit of platelets every week or every 2 weeks. The median number of platelets was only 1 unit. Indeed, 1/3 of patients needed growth factors such as GCSF, but the median number of doses of growth factors, again, for patients on the imetelstat arm was only 4. So it's not like you are making these patients need, GCSF forever. It's a very limited period of platelet transfusions and growth factor use. And importantly, the response is actually isolated from those neutropenias, meaning that you are making the patients transfusion independent. As you can see here, 40% achieve transfusion independence with with imetelstat, but at the same time they are also platelet transfusion independent. So you are not making them needing transfusions, and you are making and they are also not needing growth factor support, suggesting that they are not deeply neutropenic. So I think this is very important, that the benefit that you get by making the patient red blood cell transfusion independent is not lost by making the same patient neutropenic, needing growth factors or needing platelet support for deep thrombocytopenia. And the last point I made at the beginning is that I think it's important to look at responses in lower risk MDS, not only by transfusion independence, because this can be affected by physician behavior as well as patient wishes, but also by looking at mean hemoglobin rise. And you can see many of those patients who achieve hemoglobin transfusion independence also achieve the mean hemoglobin rise of 1.5 grams per liter, again, confirming the biologic activity of this agent. So I think the lower risk MDS field has seen very important developments. However, there are still many important questions. How do you sequence these drugs? What is the activity of imetelstat after luspatercept? Because luspatercept now is approved in the frontline setting. If we use imetelstat after luspatercept, how well does it work? We still don't know. We also don't know the impact of using combinations, because there's a lot of interest in exploring different combinations. What is the position in RS negative patients? Should luspatercept be used in all of those patients in the frontline setting? I think I'm very interested to understand the activity of ESA after luspatercept, but my current practice is to use luspatercept regardless of ring sideroblast, maybe except in patients who have an EPO level of less than 100 and very limited transfusion need, where those patients might still respond to ESA. But the vast majority of patients in the RS negative setting, in the frontline setting, should get luspatercept, in my opinion. But also we could try to aim more ambitiously, try to resolve the anemia, and maybe treat earlier in the disease course, as I mentioned, maybe even considering treatment of CCAS patients, and ideally at one point have better biomarkers for response. Now, moving to high-risk MDS, which unfortunately has not seen the same therapeutic success like lower-risk MDS, but we continue to work hard. The main approval in the last year has been the use of IDH1 inhibitor ivosidenib in the second-line setting in patients who have IDH1 mutation, but that's a small number of patients, around 3%. The reason why we are still trying beyond the standard of care, which is hypomethylating agents, is that the outcomes with hypomethylating agents by themselves continue not to be particularly great. The median survival is in the range of 16 to 18 months, and I think this is something that certainly need to be improved. We have tried to improve a lot, but as you can see, many drugs that we have added to HMAs, they initially showed good data, but once you have gone to the randomized phase 3 trials, the trials were negative. I mentioned to you that the response criteria might be one of the reasons why some of those drugs probably should not have even moved from the first phase trials to the randomized phase 3 trials. But I think we clearly need better drugs. I think that's also obvious. And I'm going to talk about two important negative phase 3 trials that were presented in EHA. But I'll start with the positive news, which is ivosidenib, which is already approved in the US in the relapse refractory setting for IDH1 mutated MDS, with very impressive data, lasting responses, CR rate of around 40%, and a very long duration of response and OS. But I think there is actually a lot of excitement about using the drug in the frontline setting based on the IDIOM trial, which came from France. This was presented by Dr. Sepere from Paris. And you can see here that in the frontline setting, this is in the refractory lab setting, which confirmed the activity of the drug with a median OS of 9 months and high response rate, as you can see, overall response rate of 64% as a single agent after HMA failure. But I think the most important results from this presentation were in the frontline setting, where the use of ivosidenib as a monotherapy was associated with CR in 48% of patients, overall response of around 80%. You can see in the Swimmer's plots durable responses, and the median OS and duration of response were not used at this analysis. So all of these data are actually quite impressive, suggesting that this drug in the IDIOM trial can be used as a single agent without HMA therapy. But I think this is a single arm, relatively small study. Of course, the challenge is that the IDH1 mutation is still quite rare in MDS, is only around 3 to 5% of patients. So while this is important, it's not going to apply for most patients with MDS. Now, moving to the two negative randomized studies, which I think have important lessons. Both of them are looking at immune checkpoint inhibitors. I'm showing you here the first randomized clinical trial using an immune checkpoint inhibitor, which was durvalumab, PD-1 inhibitor in MDS. And we know those immune checkpoint inhibitors have led to improvement in survival, and they are widely used in solid tumors and some blood cancers, such as certain kinds of lymphomas. But as you can see here, we have not seen a lot of success in myeloid space so far. However, some of the novel agents outside of the PD-1, PD-L1, CTLA-4 might have better kind of luck. So we have looked at sabatolimab, which is an anti-TIM3. So TIM3 is an immunoregulatory receptor that's expressed both on T cells as well as in the blast. So drugs that hit this receptor might not only activate the immune system, but might directly hit the leukemia stem cells which express this receptor as well as the blast. And early phase data of this antibody with the azacitidine have been promising. However, a randomized phase 2 trial, which we presented in ASH 2023, and this was published subsequently in Lancet Hematology. This was a randomized phase 2 trial that randomized patients to receive HMA with sabatolimab versus HMA with placebo. 127 patients were enrolled. Many of them had TB53. Around 1/3 of those patients had TB53. And while the primary endpoint was not met, there was a trend for improvement for the progression-free survival, and the durability of the response for those who achieved CR was double that in the combination compared to monotherapy. So based on this, there was some interest in seeing the results of the phase 3 trial, and this is what was presented in EHA. We presented these results of the randomized stimulus MDS2, which enrolled 530 patients to receive AZA with sabatolimab versus AZA with placebo, with a primary endpoint of overall survival. Again, a very large study, more than 30 countries, 150 centers. It actually enrolled ahead of schedule, which again speaks, I think, to the appetite of enrolling patients on frontline high-risk studies, because there's significantly a recognized understanding that the outcomes with HMA monotherapy are not great. So this was the primary result, an overall survival that was not statistically improved, but there was a significant trend with a median OS of 22 months compared to 18 months. So an improvement of 3.8 months in favor of the combination. Clearly, I think this is very exciting because this is the closest we ever got into improving survival, but the p-value was unfortunately 0.08. We have not been in a situation where we were this close before to an OS improvement in a randomized phase 3 trial in a high-risk MDS setting. I would note here that the control arm did not underperform. So the control arm had 18 months, which is what you typically see with HMA monotherapy. So it wasn't a poor performance of the control arm that made these results look good. However, we could not identify any specific subgroup by clinical, laboratory, or molecular features that seem to benefit from this drug, including patients with TB53. But in addition to the overall survival trend, we saw good trends in favorable trends in favor of the combination with in complete remission, in leukemia-free survival, and progression-free survival. And the toxicity profile was very similar to what was seen earlier. This drug is relatively safe to give. The myelosuppression is minimal. There was a mild increase in hematologic adverse events, but there was no significant increase in fatal early toxicity or fatal outcomes. So I think it remains to be seen what happens with this program. The sponsor, Novartis, has discontinued the development of this program, but I think there is still a lot of work undergoing to try to understand whether there is a way to salvage the situation and move the drug or other kind of agents in that space forward. The other phase 3 trial was very kind of underwhelming compared to the stimulus, because this enhanced trial, which looked at magrolimab, similar design, similar number of patients, more than 500 patients, were randomized to receive magro and AZA versus AZA and placebo, with a co-primary endpoint of CR and OS. However, this trial was stopped at interim analysis due to futility. And you can see here that there was no difference in complete remission rate. But as you can see, there was actually a detriment in the survival. It wasn't statistically significant, but the OS was around 16 months compared to 18 months with AZA alone. And you can see here that the control arm had the same OS like the stimulus trial control arm, which is around 18 months. So here the survival was clearly worse, and there was a lot of early toxicity related to the anemia, which led to this negative data from this trial. However, I think the CD47 pathway is still viable. There are a number of agents that are being studied. We presented, for example, results from a phase 1 study of this agent that works on the SERP alpha, which is the other side of the CD47 pathway. CD47 is expressed on the plasma, SERP alpha is expressed on the macrophages. So this drug actually is is an antibody that has both SERP alpha blocking activity as well as a CD40 ligand activator. And in this small phase 1 study, which actually had a good number of AML and high-risk MDS patients, many of them had TB53 mutations. You can see a significant number of patients achieve CR around 40%. and there is no evidence of anemia with this particular drug. So I think it's relatively safer to use than magrolimab, and therefore this trial is being right now expanded, and I think we are hoping to present additional results from this compound in the near future. I would finish on venetoclax. We are still waiting for the randomized phase 3 trial, but venetoclax is certainly active in the refractory setting, as we have shown in the past, strategy to azacitidine after HMA failure. And randomized phase 3 trial, which will answer the question in the frontline setting, a very large, more than 500 patient study, is fully conducted in the US, where patients were randomized to receive AZA and venetoclax versus AZA and placebo. And the primary endpoint is expected to hopefully report later this year or early next year. This trial, I think, will be very important in showing how venetoclax should be used in patients with MDS. At the end, I would conclude by, despite the failures in the high-risk MDS space, there are a lot of active agents that are being studied. Another phase 3 trial is looking at tamibarotene, which is a selective RARA agonist. This is a trial that randomizes patients who are RARA overexpressors to receive this drug in combination with azacitidine versus azacitidine and placebo. But there are other agents that are being studied, other CD47 targeting agents, as well as IRAK inhibitors, and a number of immune mechanism intervention for patients. So at the end, these are my acknowledgements and my contact information, and I'm very happy to take questions.
Thank you very much, Amer, for really a fantastic overview. And I think you did a great job in 45 minutes or so to give us really an amazing tour of this rapidly evolving field. And it's quite impressive to see. 30 years ago, I remember at medical school, MDS, other than transfusion, nothing else to offer. And now we see all of these international trials, but also drugs being approved. So we have a lot of questions. So let me try to structure them. First of all, what is your definition of complete remission when you treat an low-risk MDS?
Yes, I think this is a good question, and this comes to why those criteria were somewhat unified. Because in the past, I think complete response had a very strict definition that's not easy to get to, which required that the blast count goes to less than 5%, but also you have to improve your hemoglobin to 11, platelets to 100, and the neutrophils to above 1,000. When you compare these response criteria to AML, actually they don't care about hemoglobin there. So you could still be transfusion dependent for hemoglobin, but achieve the platelet and neutrophil recovery and still be a CR. There is actually a very nice study in high-risk MDS where if you do the AML response criteria, the CR rate is 40%. When you do the MDS criteria, it's 20%. So it didn't make a lot of sense, because we know these diseases are on a continuum. If you have 19% blast, you have 20% CR, but 40% if you had 21% blast at baseline. It just doesn't make a lot of sense. So there was an interest to try to uniform these criteria, but also account to the idea that it's not always all or none. It's not that you are either complete response or nothing. The problem is that marrow CR, which is no count recovery and reduction in blast, was mixed with this, and you have these phase 1 trials telling you, I have 70% overall response rate. Everybody gets excited, the companies start phase 3 trial, but when you look deep into these data, you find that 40% of those 70% were just blast reduction, which, as you know, we can give chemo and reduce the blast, but doesn't help those patients. So I think the introduction of these new response categories try to recognize that improvement in the platelets or in the neutrophils separately to meaningful levels combined with the blast reduction is clinically meaningful. And I think the validation results that I just showed from the Austrian group, from the Astertane group, all point out that those kind of additional CR categories carry some value. But of course, I think the best way to know for sure is to include these in big prospective randomized trials, I think, where we will fully understand those the value of these responses.
Thank you very much. This is quite pragmatic, and I fully agree with you. We have a few questions about luspatercept, and for instance, uh, for how long you would give it before evaluating effectiveness? Can it be combined with ion chelators? And are there any predictors of response and efficacy?
Yeah, so all of these are, I think, very good practical questions. I think for the first question, it's actually we don't have like very good predictors, but the good news is that it has like a wide indication in the frontline setting at this point. It does work in most of the patients, as I showed, like the most recent update which we presented in ASCO 2024. Actually, the primary endpoint on the luspatercept arm was met in 70% of patients. Transfusion dependence for 12 weeks combined with a hemoglobin rise. So in my opinion, you can give it really to any patient in the frontline setting. The only situation where I think is a predictor or potentially is unlikely to respond is if the EPO level is more than 500. That applies to most drugs. We actually don't have a lot of data because almost all the trials with luspatercept excluded patients who had an EPO level above 500. So I'm interested to see some additional data in that setting. In terms of combining it with iron chelation, certainly is done. We do it all the time. I don't think there is any concern there. I think iron chelation in general is not easy to give for this group of patients because they tend to have a lot of GI side effects and there's a high rate of discontinuation. But generally, if you have improvement in effective erythropoiesis and patients stop needing transfusion dependent, I think many of them, the iron overload issue becomes less prominent. But there is no problem in using both combination. I think your last question was related to the dosing and when do you hold. So generally, actually, this has been studied in a very systematic way within the trials. So if you start with the 1 milligram dose currently, and after 2 doses, and the drug is given every 3 weeks. So after 6 weeks, if you're still needing blood, you go up to the 1.33. The same thing happens after the next 2 doses. And once you go to the maximum dose, which is 1.75, you give it for 3 doses. If there's no clinical benefit, and here I would emphasize no clinical benefit, not transfusion dependence. So as I mentioned, significant reduction in blood transfusion need is a clinical benefit. Then you discontinue the drug. What I showed is that we are actually doing the reverse trial where you are starting with the maximum dose from the get-go, because what we know is most patients have to go through this dose escalation to get the benefit. And what happens in the community, because it's a relatively easy drug to give, sometimes doctors just forget the patient on the small dose and never escalate. So I think this is important to remember to escalate the dose, which is the same thing with EPO. We see many patients go on smaller doses and never escalate.
Excellent. Thank you very much. That was very clear and very practical. We have a few questions actually similar to the luspatercept story, but now about imetelstat. And by the way, congratulations, because I think you played a key role in bringing this drug to approval. So are there any predictors of efficacy, and for how long should it be given, and any advice on practicality of using this drug?
Yeah, and again, I think those are important questions. I think in terms of like predictors for response, we have not also identified any specific features that predict for response. So this drug actually works regardless of the EPO level, even in patients who have more than 500 EPO level. It works regardless of whether you have ring sideroblast or not. We even looked biologically at things such as the telomerase activity and the telomere length, etc. And there were like no specific like highly predictive features of its working or not. So I would use it basically in the second line setting, regardless of those aspects. I think, as I mentioned during the talk, one of the important aspects we need to understand better is how well does it work after luspatercept, because this has not been fully kind of understood in previous trials, because the Amer trial only had 7 patients who had previous luspatercept. So I think this is very important as luspatercept moves to the front line setting. In regards to the hemoglobin rise and the thrombocytopenia, this is actually a common, I think, issue that came up during the approval discussions during the ODAC discussion, Oncologic Drug Advisory Committee of the FDA. And there was a very robust discussion about the value of transfusion independence, but also about the thrombocytopenia and the neutropenia. And this is why I think the abstract that I presented in ASCO 2024 was important, because it noted two things: that the patients who respond, the median transfusion need is for platelets only one. And also, most of those patients will not become transfusion dependent or need growth factor continuously. So it's not that you are making the patient red blood cell transfusion dependent and then you are making them at the same time platelet transfusion dependent. This is a very important concept to get. In terms of long-term thrombocytopenia, I think it's always very tough to kind of disconnect what is disease progression, what is drug. But with the drug itself, we know generally that within 2 weeks, if you hold or delay the dose, most of the patients, their platelet count will improve. So we are not seeing those like long refractory protracted thrombocytopenias with the drug.
Excellent. For the sake of time, we'll take only one additional question, and this is about sabatolimab. And do you believe that there are specific genetic subgroups who are more or less able to respond to this new agent?
Yeah, this is an outstanding question. And since we have seen like the results of this study, which, clearly you can imagine how disappointed we were, because it's, an OS of 3.8 months, but the p-value was only 0.08. And I think the real question in my mind is this, there are really two possibilities. Either this is the real effect that the drug is active, or is this somehow a statistical chance? And but I think p-value of 0.08 compared to 0.05, I mean, generally the way you think about these p-values is that what is the chance of something happening by chance if you conduct the trial 100 times? And this is what p-value of 0.05 means. It's less than 5 times out of 100 here. So it's 8 out of 100 that this is by chance. So my own belief is that this is not just a chance effect. But then is the effect, is it across all the groups, but just weak enough that we did not enroll enough patients? You are a top expert in myeloma field, and I think many of your trials tend to have 700, 800, sometimes thousands of patients. This happens in solid tumors all the time. So I kept wondering, if you use a bigger trial, if you had a 800 patient rather than the 500 who were in the trial, would that p-value have been significant because you have a like enough power? But the point that the question raises is very important. There could be biologic subsets that are benefiting, and just you are diluting the effect by including all patients. I make the analogy to the IDH inhibitors. If you test them in all patients, regardless whether they have IDH1 mutation or not, probably not going to be clearly positive. So that work is still ongoing, but so far we have not seen specific biologic or genetic subgroups that seem to derive benefit more than others.
Fantastic. Thank you very much, Amer. That was an outstanding summary and webinar. You're watching the International Academy for Clinical Hematology around the globe and across the hematology field. Thank you for being loyal to our activities, and see you soon.
Thank you so much.